So now that we already started comparing proteins such as chitin found in exoskeletons of insects or other arthropods, such as crustaceans and arachnids, as well as the cell walls of fungi, so two very different groups that share the same chemical polymer, that you still have to find out, is this a protein, is this a polysaccharide, what is chitin? So we compare that with keratin, which is the most abundant protein, or one of the most abundant proteins in skin and feathers and fur and nails and hair and so on. How else can we use biochemistry, since now we have the tools to compare different chemicals in order to determine phylogeny? So if organisms share the same chemicals, they could have morphologically, they could have come from the same group, or they could have come across making that particular chemical by chance as well. So one example is caffeine. Even though caffeine, it's found in the seeds of the coffee plant, it's also found in a lot of other plants, and the same with aspirin. Even though salicylic acid, it's characteristic to willows, it can be found in numerous other plants, flowering plants, that are not related or not very closely related to caffeine, to coffee trees or tea plants and so on. So I want you to take a couple of minutes and come up with other examples that you can think of that are comparing different biochemicals in the pursuit of phylogeny hypothesis. So, for example, even humans have a particular scent that could be genetically controlled. So you might stink more with this side of the family or the other side of the family, for example. Maybe you find out much more exciting biochemical examples. But two main chemicals that are very useful to comparing biochemistry in the pursuit of determining phylogeny are comparing DNA or protein sequences. And one very ingenious way of using DNA comparisons for phylogeny is to produce molecular clocks. So what is that referring to? If two groups have similar successions of the nucleotides, then they will be more closely related. So it turns out that if we look at the whole genome of humans, we are about 97 to 98% similar to chimpanzees and bonobos, but we're also 88% similar to mice, and we're about 73% similar to zebrafish, and we're even 28% similar to rice. So our genome shares 28% with rice, okay? So how do we create, how are these moleculars, how do they work? What do they refer to? Well, we already established that mutations accumulate at a constant rate. And most mutations are neutral. So even though they are mutations that could be beneficial, rarer, or detrimental, sadly more common, because it's easier to destroy a good sequence that codes for a particular protein versus inventing or coming across a new protein that is beneficial. But many of them could be neutral. And when they are neutral, we can use them to produce these molecular clocks. And why in particular neutral? So why wouldn't we look at the beneficial mutations or detrimental mutations? So please take a minute, turn the video off, and speculate. How are neutral mutations more useful to us to determine when exactly two groups, two sister groups at a node in a cladogram have differentiated. And it turns out that since they're neutral, it means they are not exposed to the visibility of natural selection. So natural selection wouldn't eliminate it if it's detrimental, or promote it, or facilitate its multiplication in future generations if it's beneficial. So it's more useful for us to use neutral mutations to determine when exactly two groups have differentiated.